So one of them is that protein carries the genetic information, and the other hypothesis is that DNA, the nucleon now called DNA, carries the genetic information. So, you know, scientists did not know which one of these. There was these two schools of thought that, oh, it's protein that carries the genetic information, and then no, it's DNA that carries the genetic information. And so based on their hypotheses, there are two alternative hypotheses in their experiment, we are able to know which of these two molecules does actually carry or transmit the genetic information. So how do we know that? Well, let's review their experiment. Okay. So first we have viruses, right? Okay, viruses. Okay, so here's what a bacteriophage kind of looks like. Okay, they kind of look like little, I don't know, aliens. But viruses are composed of two things, two molecules. They're composed of protein and they're composed of DNA. Okay, that's all that makes up viruses. That's it. And what viruses do, in order for viruses to reproduce, they have to get into a host cell. They can't reproduce on their own. So like when you get the flu, okay, those flu viruses are getting into your respiratory cells. And they're using your respiratory cells to reproduce more virus. So what viruses do, specifically they study bacteriophages, is they attack a cell. So this is a bacteria cell, okay? And what they do is they land on the bacteria cell and they inject something, okay? They inject something. And this something that they inject takes over the host cell, hijacks it, and causes the host cell to produce new virus particles, which then they will then assemble into new viruses, and then the cell bursts, releasing the new viruses. So the cell gets destroyed in the process. Okay? So that's the life cycle, or the cycle of how these viruses infect cells and hijack the cell to become, to replicate more viruses. And so Hershey and Chase were like, well, what is it that the bacteria is injecting? Are they injecting protein or are they injecting DNA? And if we can figure that out, then that should tell us, you know, what transmits genetic information. So they did this really cool experiment. So how did they set up their experiment? What did they use? Okay, because obviously viruses are too small for us to see without a fancy, you know, expensive electron microscope. So how did they do this? Okay, they had to use something in order to track this. Okay, so what did they use? What did it tell you that they used in the clue set? Okay, yeah, they used some sort of marker. Okay. So they use these, what we call markers, and these types of markers, radioactive markers, are used in medical research still to this day. You know, if they're trying to find a blood clot, or they're trying to see if a tumor, locate a tumor, or has a tumor metastasized and spread, they'll use these radioactive isotopes. You know, they inject it, and it clings to the whatever you want to see, and then when they take a scan, like an MRI or something, it glows. Okay, so these radioactive isotopes we can use as tags or markers. So that's what Hershey and Chase did. Okay, Hershey and Chase did. So they had two setups, right? So hypothesis 1 is the protein, and hypothesis 2 is that it's DNA. So okay, we got to have a virus, same type of, okay, bacteria, but in this case what they did is they took these tags, these radioactive tags. So I'm going to use pink for the protein, okay, and the protein marker attaches to protein. The DNA, I'm just going to use the color green. Okay, and so the DNA marker is going to be green. So they have these side by side, okay, these setups going. So if we assume that it's protein that transmits the genetic information, then what do you predict? Okay, predict. What would we predict we would see on the inside of the cell here for what the virus injects into the cell if it's protein that carries the genetic information? What should we see on the inside here? Okay, we should see what color? Pink. Okay, we should see pink. If that's what, okay, if that's what is being transmitted. But what if it's DNA that transmits the genetic information? Okay, then if it's DNA that carries the genetic information, what would we predict we would see on the inside of the cell? What color? Green. Okay, we would predict that we would see green. Okay, that's our prediction. Okay, their actual results. So the actual results when they ran these experiments. So their actual results for both sides, what they found is they found okay, so based off of the actual results from both sides, what conclusion can we draw? Which hypothesis is supported? Hypothesis 1 or 2? Okay, hypothesis 2. Because what's found on the inside, what glowed on the inside was the green, okay? Over here, there is no green marker. There's only the pink marker, which was only found on the outside of the cell. So that told them that, oh, the protein marker does not enter the cells. The DNA does, because the DNA marker is what was found inside all the cells on this side, okay? So from that, they drew the conclusion that it is DNA that carries the genetic code, not protein. So all of this is what we know based on the information, the discoveries presented to you in ClueSet 1. So we know that there are chromosomes that consist of protein and nuclein, which is the DNA. Okay, I'll just put DNA here. Okay, that the sperm only contribute. Their basic contribution is just chromosomes, and that the eggs, okay, and the sperm, the gametes, have half the amount of chromosome in them that all the other body cells do. And we know that now that we could say pretty with confidence that it's DNA that transmits the genetic information, okay? So now that scientists know this, now they're going to focus more on how does DNA transmit the genetic information by looking at its structure. And so that was what went forward with the scientists looking at doing their research going forward. So now we're going to kind of play around the models that you have in front of you. Those little pieces are showing the structure of the DNA molecules, okay? So those are there. So now we're going to look at ClueSet 2, okay? And ClueSet 2, what I want you to do with ClueSet 2 is, again, read it, and then using the model pieces, try to arrange only based on the information from ClueSet 1 and what you're reading in ClueSet 2, try to put together what you think a basic model of DNA might look like. Okay, and so you give about 10 minutes to do that. And you'll want to float around because they're going to probably want to put together, oh, the DNA molecule with the bases on the inside. But ClueSet 2 is the picture from Rosalind Franklin and a little description of what it tells you. So float around, and say, well, if they're starting to put the bases on the inside, you might say, hey, how do you know that? Where did you read in the clue sets that those go on the inside? And if they're like, well, I just know, then remind them that, again, we need to go with only what is presented to us in ClueSet 1 and ClueSet 2. Okay, so arrange your models there. Okay, then have a short discussion about ClueSet 2. So